The voltage across the inside to the outside of our membrane is normally about negative 90 millivolts. It's not very big, but it's definitely there. When it opens and the positive sodium rushes in, we're going to go from negative 90 to positive 75. So we're going to completely flip the charge on our membrane. We're going to go from negative to positive. And that's what's going to have a whole bunch of effects in our cell. Sodium rushes in, we go from negative 90 to positive 75, but then potassium is going to go out and take the positives with it and bring our membrane potential back to negative 90. It's going to happen very, very quickly. This is going to open, sodium rushes in, potassium rushes out, and you're done.
This is happening every time you move a muscle?
Every time you move a muscle. Yep. And so, this is a synapse here. So this is that receptor that we just saw on the last slide. So this is going to open. Sodium's going to come in. But one membrane opening and closing is not going to create a big enough change to make anything happen. And so what's actually going to happen is it's going to trigger a whole bunch of other pores to open up. So these are voltage regulated gates. And so when this opens, the voltage right here is going to change. In the big picture of the cell, it's almost insignificant. But when the voltage changes right here, some of it kind of moves over to here and opens these voltage regulated gates. So voltage regulated gates open when the voltage around them is what it's supposed to be for that to open. There's a certain set of conditions for a voltage regulated gate to open. If the voltage here changes, they're going to open. And when these open, there's a whole bunch of these. So now you're going to have an entire flood of sodium that's going to come in and cause the membrane potential to change even more. And so when this one opens, it's going to cause the potential here to change. And so then there are more and more and more and more. So over the entire surface of the muscle, all of these pores are going to open up and a whole bunch of sodium is going to pour in. So now, we're going to move on to the second step, the excitation-contraction coupling. So, we have this action potential. The action potential is that moving of the membrane potential change over the membrane. And so one pore is going to open, it's going to cause the next one to open, and the next one, and the next one, and the next one, and the next one. And this positive charge is going to move slowly across the length of the muscle. So that is our action potential. And when that sodium comes in, it's going to end up in the T tubules. And the T tubules are connected to the sarcoplasmic reticulum, which has all of the calcium in it. When the action potential reaches the T tubules, it's going to open channels in the sarcoplasmic reticulum, which was going to release calcium. The calcium can then come on and bind to the troponin and remove the tropomyosin and allow the whole contraction to happen. So this is that coupling. So we had the excitation here with our axon opening that acetylcholine pore. Once that opens, now we have the coupling. It's going to cause the membrane potential to move down the T tubule into the sarcoplasmic reticulum, releasing the calcium. Going from the neuron to the release of the calcium is this coupling step. Calcium is going to come in. It's now going to bind to the troponin. When the troponin binds calcium, it moves. And so it just shifted kind of towards the center a little bit, and now we have exposed these active sites. They're little dimples perfectly shaped for the head of a myosin molecule to grab on. So, with calcium bound, tropomyosin move, the myosin head will now bind. And now we can contract. And so, the head of a myosin molecule is able to flop around. It's also able to take ATP and convert it into ADP. So remember, ATP has high energy. That's what's your energy currency in your cell. Once you use some of that energy, now it's ADP, and it's not able to be used anymore. And so when ATP binds to the head of myosin, it's going to cock it back. And so normally the myosin started like that. It's going to bind ATP, it's going to cock back, it's going to grab on to that, and then it's going to hydrolyze or break apart ATP into ADP, and it's going to flop back to where it was. And so by cycling ATP with ADP, this myosin head is going to keep going back and forth, helping to move this thin filament to the left. So the power stroke is when it releases. And so we have a power stroke when it's going to come this way and straighten out, and then a recovery stroke where it's going to cock back. So ATP brings it to the right, and then it hydrolyzes it and it moves back. ADP falls off, another ATP comes on, and it comes back. So it can just keep going back and forth and back and forth, and it's capable of doing that 5 times per second. Each time it does it, it's going to use 1 molecule of ATP. Now, imagine, remember, this is a tank. We're trying to contract these together. If this has to let go to recock, why doesn't our muscle relax every time it has to do that? Picture our big diagram of the overlapping fibers. What was on the thick filaments? We had the myosin heads, right? How many were there? A lot of them. And so just by random chance, only a few of them are letting go at any point in time. And so the ones that are still holding on are going to hold it in place, waiting for the other ones to grab on and pull, so the ones that were holding on before can let go and recock. Once we've contracted, we need to relax. And so in order for relaxation to happen, the nerve stimulation has to stop. If the nerve is still sending the acetylcholine out, we're going to continue to have this pore open. It's going to make the voltage-gated channels stay open, and the contraction is going to continue. If nerve stimulation stops, this is going to shut. And then eventually the voltage-gated channels are going to shut. And if they shut, then the calcium is going to go back in the sarcoplasmic reticulum, and the whole process is going to stop. Acetylcholine will break down over time. It's actually a fairly fast process. So it's continuously being released into here, but if you stop releasing it, what's in here is going to very quickly break down. So it's not going to take a long time for these to soak it all up. It's just going to break down very quickly, and the signal is going to stop. The calcium is going to get pumped back in the sarcoplasmic reticulum, which means the troponin is no longer bound to calcium. The tropomyosin will move back to where it was, blocking the active sites on the thin filament, so the myosin can't grab it anymore. There is a protein in the sarcoplasmic reticulum called calsequestrin. So calcium and sequester. So it's going to hold onto it in the sarcoplasmic reticulum here. It kind of holds onto it and makes sure it doesn't escape when it's not supposed to. So calcium is going to get lost. Troponin loses calcium. Tropomyosin is going to flip back to on top and block these active sites, and the whole process is going to stop. Remember the elastic fibers, springs, when the myosin lets go, they're going to pull everything back to where they were, so that if the stimulation comes again, it can contract again. So, obviously there's a connection here between the muscle and the nerves. Today we're talking primarily about the muscle, but you saw that the first step in that contraction process comes from the nerves. And so nerves are coming from our brain or our spinal cord. They're going to tell the muscles to contract. And they're going to tell units of muscles to contract at the same time. So one little muscle is not going to do a whole lot. So this is called a motor unit. It is one nerve fiber and then all of the muscles that it controls. So if a nerve fiber fires, all of the muscles that it attaches to in the other end are going to contract. And so that combination of the nerve and the muscles is called a motor unit. So the connections between that neuron and the muscle is throughout the muscle. So it's not just going to attach to one end. It's going to send the signal to one end of a muscle, and the whole thing's going to contract. It's going to branch. It's going to come out, and it's going to attach throughout the muscle and all of the throughout the length of the muscle fibers, so that the whole thing can contract at once. Each contraction is not very strong, but we have a whole bunch of them. We have a bunch of contractions within one myofibril. We have a bunch of myofibrils in one muscle fiber. We have a bunch of muscle fibers in a fascicle, and then we have a bunch of fascicles in a muscle. So we have a whole bunch of small contractions that happen at the same time. The fact that there are such a large number is what's going to allow us to maintain strength over a long period of time. It allows some of them to let go and recover, while the other ones hold everything in place. Usually, if we want to do anything important with our muscles, we're going to have to use multiple motor units. And so each motor unit is going to serve a slightly different purpose. And if I want to flex my arm like this, my brain's going to have to tell multiple motor units in my arm to move. If I go like this, I'm probably using some of the same motor units, but not all of them, and I'm probably also using some additional ones. And so multiple motor units have to be recruited together in order to cause a movement. One motor unit is usually about 200 muscle fibers, and so that's not a whole lot. That muscle fiber is a cell, remember, so 200 cells is peanuts. And so our brain is able to control very small units of muscle in order to very fine-tune our movements. If you had to move gigantic blocks of muscle together, there were only so many movements that you could do. But the finer little groups that you can choose to move together, you can do all different things, right? There's only about 3 to 6 muscle fibers per neuron. And think about the movements of your eye and your hand. The eye and the hand are the muscles in our body that have the most fine movements. I can do all kinds of stuff with my hand that I can't do with my toes, right? I can move my eyes all over the place. And I don't even really have to think about it. I can make them go left and right and up and down and spin and do all kinds of cool stuff. I can't do that with my big toe. My big toe just kind of goes like this. It's not a very limber big toe. There are also large motor units that are for strength. And so things like my fingers and my eye, it's very fine controlled, so they're fine motor, they're small motor units. But they're not going to be very strong. I can't put a weight on my eyeball and lift the weight with my eyeball. But I can with things in my arm or my legs. They're very, very large motor units with hundreds and hundreds, if not thousands and thousands of fibers. So the calf has 1,000 muscle fibers per neuron. So up here, we only had 3 to 6. And so our brain can say, fire this neuron, and only 3 to 6 cells are going to contract. But in my calf, my brain says, fire this unit, and 1,000 or more muscle cells are going to contract. It's not very fine movement, but it's very strong. So we can take this whole process and put it into a simple little graph. Okay? And so this is going to be called a myogram. So remember, myology is the study of muscles. So this is a myogram. And what we're going to look at here is on the x-axis is just time. So this is just as time goes by. And the y-axis is going to be the strength of contraction. It's not an instantaneous process, the contraction or the relaxation. It takes a little bit of time. Remember, this whole process started based on a change in membrane potential. There is a threshold that you have to meet in order for that contraction to start. A tiny little membrane potential change is not going to cause our muscles to contract. The threshold voltage is that minimum voltage. Until you hit that threshold, nothing is going to happen. Imagine when we talk about our channels, our acetylcholine receptors are going to open. They're going to let some sodium in. We're going to get a small change. But the large change in potential doesn't happen until the voltage-gated channels open. And they only open at a specific voltage. And so we said the starting is about -90. So let's say those channels don't open until we come up to -70. And so if we get a small change up to -75, it's not going to open. We have not reached our threshold. So nothing is going to happen. It's just going to sit there. Another term we're going to use is twitch. A twitch is a very quick contraction and relaxation. If I do this, this is not a twitch. We all have all seen and experienced muscle twitches, right? It's just very, very fast. It's contract, relax, contract, relax. So our strength is going to be essentially 0 here. And then we're going to hit our threshold. We're going to contract. And then we're slowly going to relax. The latent period is this little tiny gap between when we reach the threshold and the contraction actually happens. It's very, very, very small. It's nothing we could ever notice in real life. It is the time for that excitation and the excitation contraction to happen. It's the time needed for those channels to open, for the sodium to come in, for the calcium to be released, and for the tropomyosin to move. We're talking nanoseconds, right? It's very, very, very fast, but there is, in fact, a delay. And so that is the latent period. Then we have our contraction phase. That's when the filaments are sliding. Our sarcomere is contracting. And then we contract, we max out our tension, or our force, and then everything kind of dies off. The nerve signal stops, The pores are going to close, the ions are going to go back to where they belong, the elastic fibers are going to pull everything back to where we started. That's our relaxation phase. And if this is a twitch, all of that is going to happen between 7 and 100 milliseconds. A millisecond is a tenth of a second. So, I don't know what I just said. 100 milliseconds are a tenth of a second. Is that what I said the first time? You don't even know what I said the first time. So 100 milliseconds is a tenth of a second. So that a tenth of a second is the longest twitch. And so you can imagine how fast a seventh milliseconds is going to be. If we have not reached our threshold for our stimulus, there will be no contraction whatsoever. It is not a slow increase. It is no increase. We hit threshold and then it's big. It's all or nothing, more or less. When we hit threshold, we produce our twitch. The voltage that we give to a cell is not necessarily going to determine how strong that twitch is going to be. You can deliver the same voltage change to multiple muscles in multiple places and receive very different strength in contraction. Number one is how much was it contracted when you hit it with your voltage change. Remember, every time it relaxes, it has to come back to where it started. And the further they are apart when they start, the more they can contract. Imagine you contract once, and then you start to relax, and then you hit it again. It's not going to move very far the second time. If it doesn't move very far, it's not going to provide very much force. So how far and how long the muscle fiber was to start with is going to help determine the amount of force produced. Muscles are going to get tired. What do you think we mean by muscles are going to get tired? What's going to prevent that muscle from continuing to work at 100%? I mean, muscles don't sleep. So what's going to happen to that muscle, do you think? It's going to prevent it from doing the same thing over and over and over again. Run out of ATP. What else could it run out of? Sodium, potassium, calcium. That's another big thing.
Motivation.
What's going to be needed? I mean, we can make more ATP. We're going to talk more about that. But in order to make energy, what do we need? Why do we breathe? Oxygen. Remember that myoglobin? Our muscles are going to run out of all of those things that they need. And so they're not going to have this abundance of it. They're going to continuously be supplied, but at that point, they can only use as much as getting delivered to it. It's constantly waiting for to be resupplied, and just burning it as it gets to it. Also, a muscle is, yeah, go ahead.
So if I was in the gym and I hooked myself up to the breathing mask with just oxygen, would I tend to be able to go longer, or does this still have to do all its processing in order to be?
If the oxygen was what was limiting you in the first place, then yes. If the oxygen was not the limiting factor to begin with, though, then no. Things we know, warm muscles work better. That's number one, why we warm up before we really intense exercise. That's why if you're running in the freezing cold, you're probably not going to be able to run as fast as if it's a nice temperature outside until you get warmed up. How much water is in your muscle is going to influence a lot of things, including concentration of those ions. You need the water to move things throughout the muscle. But if you have too much water, it's going to dilute the entire process, and those membrane potentials are not going to change as fast as they should. And also, the frequency, like we were talking about, the frequency at which you cause these contractions is going to have a very big effect. If you do it too quickly, there's gonna be a whole bunch of little twitches. But if you do it too slowly, it's gonna come back, it's gonna sit there and it's gonna wait, and then it's gonna go. It's gonna come back and sit and wait. You wanna time it so that as soon as you get back to the full length, it's gonna contract again. That's gonna give you the maximum amount of force. Obviously, we need to be able to control how much force we apply. If I'm going to pick up this, I'm not going to apply as much force as if I'm picking up a barbell, right? If I did, my arm would just go flying. And if I tried to pick up the weight with the same amount of force that I used to pick up this, my arm's just going to hang there. I'm not going to be able to do anything. So we need to be able to control the strength. So the last slide, we said that higher voltage does not necessarily mean stronger contraction, because there are a whole lot of other factors that go into it. But at the end of the day, a stronger voltage should produce stronger contractions if everything else is held the same. Remember that there are a lot of variables here, and if you change multiple at the same time, you can't really guess at what's going to happen. If you hold everything the same, we're going to cause a higher contraction. The higher voltage is going to be able to activate more nerve fibers. If we activate more nerve fibers, we're going to have more contraction. The ability to activate multiple motor units at once is called recruitment. That's an important word. Recruitment is the ability to activate multiple motor units. And so imagine, in my bicep, I have multiple motor units. If I just need to go like this in air, I don't need to activate very many of them in my bicep in order to do this. But if I'm going to pick up something very heavy, I'm going to have to activate all of the motor units that I have at my disposal. And to do that, I'm going to send a stronger voltage. Stronger voltage coming from my brain into my bicep is going to cause more of them to snap into action. Generally, a weak stimulus is going to recruit small motor units. It's going to require a large stimulus, a large voltage change, in order to activate large motor units. And so if I just need to go like this, my brain doesn't need to send a very big signal. And by sending a weak signal, it knows that only the small motor units are going to turn on. It doesn't even have to worry about knowing, say, I want this one and this one and this one. It can just say, send a weak signal. I know only the small units will activate. And I know which ones are the small ones. If I want the big motor units to activate, I'm going to send a big signal, and so that all of the motor units are going to activate and provide maximum strength. So again, we have time down here, and on our y-axis is going to be strength. So here we have a contraction, a relaxation, and then we're going to wait a long time before we contract. And so by the time we contract the next time, we have lost most of our force. But if I contract, I wait for it to relax, but I wait for it to almost completely relax. It's not completely relaxed, it's almost completely relaxed. And then I twitch again. I'm able to come up a little bit higher than where I started. It's like if I contract and I come back to here, I'm able to produce the same amount of force the next time as if I retracted to here. If you're almost all the way back, you can still get 100% of the force. But the further out you come, the more force you have lost from the previous contraction. And so essentially the way it works out is this amount of time gives you the maximum contraction with the minimum amount of relaxation. So by doing it over and over and over and over again, we're able to build on top of each contraction. And so the amount of force that we're applying over time is actually going to go up. But there's a maximum amount that we can contract. And so you can see it's kind of coming up to a maximum here. Each contraction is getting smaller and smaller and smaller and smaller. And so eventually, this contraction is going to completely go away. We are at maximum force in the contraction and relaxation is essentially completely invisible. We are permanently contracted. This is called tetanus. This is tetanus when we're completely contracted. The ability to build on top of each other is called trepi. Tetanus and trepi are two important words. So trepi is the ability to build one contraction on another to provide more force. When you max that out, that is called tetanus. For the most part, this is not going to happen in real life. If you take a muscle in the lab and you give it a very, very strong electrical current, or you put a whole bunch of calcium on it, this can happen. Where do you know that tetanus does happen? When you're tased? I guess that makes sense. I've never heard that. But why do you think that happens? If I get tased, why am I going to go into tetanus? Why? Why are they all flexed at the same time?
They're overloaded with electrical charge?
Yeah. Normally that charge comes from my brain. But now it's coming from taser. And so that whole electricity is going through my body, and that's a very, very strong stimulus. So it's going to make all those muscles fire at the same time. Yeah, go ahead.
Would a seizure also be like really tense muscles?
I think don't quote me on that. I think a seizure is essentially an overactive brain, right? So it's sending these signals over and over and over again. And so it's not supposed to be contracting, but the brain is gone haywire and is telling it to contract. What's another very, 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 very easy situation where you might have tetanus?
Charlie horse?
Charlie horse. What?
When you're in cab.
Yeah, I don't know exactly what Charlie horse is, so I don't want to say yes or no.
Bacteria infection?
Yes. And what would that infection be called? If you went to the doctor, you would say, I think it might, well, you wouldn't say.
Well, tetanus.
Tetanus, right? Tetanus is a thing.
Rusty stuff.
Yeah. And so there are bacterial infections that will cause this muscle contraction to happen. And so it's called tetanus. Essentially, your jaw locks up. The muscles on your jaw are in tetanus. They are locked. I mean, they are really, really, really tight. If somebody's trying to pull your mouth open and they don't do it right and they get their finger stuck in there, they're probably going to the hospital with their finger in your mouth so they can give you a muscle relaxer to get their finger out of your mouth. That is, of course, assuming that part of the finger stays in the mouth and while the other half does not. And so then you're both going to the hospital for different reasons. But it's very, very, very strong. Okay. So now we're going to take a look at it big picture. And so moving my muscles, okay? Getting away from the cellular level and getting up to the body level. And so obviously, I could do multiple things if I pick something up. If I hold this in place, are the muscles in my arm changing length? No. When I went from here to here, they changed length. But now they're not. If they completely let go, though, my arm would flop back. And so they are producing force. They're just holding where they are. So the myosin heads are grabbing letting go, grabbing and letting go. And so it's kind of timed so that when one grabs, another one lets go. And so you're not adding any additional force. And imagine all of us held up some big, heavy box. There's about 20 of us. Let's say we only needed 18 of us to hold it up. Certainly if we have 20 of us, we can hold it up. And we can actually take breaks. As long as no more than 2 people let go at once, we can keep the box up. The box isn't going any higher. The box isn't falling to the ground. As long as we have at least 18 people, it's going to stay in place. This is called an isometric muscle contraction. So iso means same. And so think metric is meaning measurement. So the measurement of your muscle is going to stay the same. And so the tension in her muscle is staying the same while the length stays the same. Usually, if I am going to pick something heavy up, so let's say I have a heavy bucket of water, and I reach down and I put my hand on that handle, if I just jerk it up, it's not going to work very well. The very first thing I'm going to do, I'm going to pull it until my arm gets tight. And I say, okay, it's no longer easy to lift. Now I'm going to apply a whole bunch of force. So a lot of times an isometric contraction is going to happen before the contraction is going to change our length. This is also what's important for our posture. I'm standing here. I'm not wobbling, at least not from my perspective. I'm not falling over. My eyes aren't rolling around in my head. The muscles are holding everything in place. If they let go, I would flop to the ground. They're constantly doing these isometric contractions to hold everything in place. Isotonic is going to be where the length is actually going to change. And so tonic is referring to tension. So again, iso is same, tonic is tension. And so when she lifts this weight, the tension in her muscle is going to stay the same, but the length is going to shorten. When she lifts it up, her muscle is going to get shorter. When she lets it go, her muscle is going to get longer. So we have 2 types of isotonic contraction. There's concentric contraction, is when the muscle shortens. So that's going to be lifting up. There's also eccentric contraction, is when you slowly let it down. You're not going to lift a heavy bucket and then just let it go. You're going to very slowly let it down. So the tension in your muscle, as you let it down, is going to stay the same. But as it's staying the same, the length is getting longer and longer and longer until you're going to set that bucket down. So this is just another fancy looking graph. In reality, it's very, very simple. We have 2 lines. The red one is going to be the length, or the tension on your muscle. I'm sorry, it's white right there. It says muscle tension. And so this is muscle tension. And then the purple line is muscle length. And so before I start contraction, my muscle is as long as it's going to be. As the tension goes up, the length is going to get shorter and shorter and shorter until my muscle can get no shorter and my tension can go no higher. It's just as one goes up, the other one goes down. It's just an opposite relationship. Okay, so that is the contractions. Now we have to talk about the energy that goes into it. Because, remember, we're doing 5 contractions per sarcomere per second. And we have a whole bunch of them. And each one of those contractions is going to require 1 ATP per myosin molecule. So that's a lot of ATP that we're going to require. yes, we have ATP in the muscles to start. But like we said, we're going to run out quite quickly. And so the ability to resupply our ATP is going to be based on the oxygen and then the nutrients that we can burn with the oxygen in order to get that ATP. Very fast energy is going to come from glucose, but long-term storage in our body, remember, is some things like fatty acids and glycogen. So we can make ATP two different ways. There's anaerobic fermentation and aerobic respiration. We're not going to talk too much about these, just enough to understand how the muscles work. This is a big section in 2086, I believe. And so we're not going to go too in-depth into this. But anaerobic fermentation does not use oxygen. Aerobic uses oxygen. Think of aerobics. That's going to use a lot of oxygen. Anaerobic means not aerobic. So this is going to happen without oxygen. This will require oxygen. So anaerobic fermentation is going to take glucose. It's going to give a little bit of ATP, not very much, and it's going to convert that glucose into lactic acid. And lactic acid causes your muscles to stop working. And so as the lactic acid builds up, your muscles are going to get tired and they're not going to work very well. On the other hand, aerobic respiration is going to take the same glucose molecule, make a whole bunch more ATP. It's not going to make any lactic acid whatsoever. But as soon as you run out of oxygen, this does not work at all. And so we need to have both going on. In a perfect world, we would only ever use this. But it's not a perfect world. As soon as you start running, your muscles are going to run out of oxygen. And so we're going to switch over to anaerobic fermentation and then supplement that with aerobic respiration as fast as our blood can supply new oxygen. So eventually we're going to have both of these going on at the same time. And so our little stick guy is going to start running. And as soon as he starts running, for about the first 8 to 10 seconds, he has enough oxygen in his muscles to run using aerobic respiration. But after about 10 seconds, or after about 8 seconds, he runs out of oxygen. He's going to run out of easily accessible oxygen, and he's going to have to access kind of a reserve of oxygen in what's called the phosphagen system, which we'll talk about in the next slide. But that's only going to last him maybe another 10 seconds. So he has 10 seconds of easily available oxygen, 10 seconds of oxygen in reserve that he can take out, but then he is toast. He's out of oxygen. He can only use the oxygen that's coming constantly through his bloodstream, but that's not fast enough. So now he's going to switch over to the anaerobic fermentation, and that's essentially what he's going to use until he finishes running. But as you're using that anaerobic fermentation, you're going into what's called oxygen debt. You cannot allow that lactic acid to sit around. You're saying essentially, okay, I'm going to make this lactic acid now, but then when I stop running, I'm going to breathe a whole bunch to get in the oxygen that I should have had when I was running to get rid of that lactic acid and pay off your debt. So that's why when you stop running, you don't stop breathing hard. As you're running, you're breathing hard, trying to do as much of the aerobic respiration as possible while you're running, but then you finish and you have to pay back your oxygen debt. As you breathe and breathe and breathe, the lactic acid will go away, your debt will come back to zero, and then you'll start breathing normally again. So, this says short exercise is a 100-meter dash is going to be with oxygen. And so based on this, this says easily available oxygen is for 10 seconds. So if you can run the 100-meter dash in 10 seconds, then I suppose then you can do it with complete aerobic respiration. I don't think any of us are going to run a 10-second 100-meter dash. So we're going to start running. We are going to take the oxygen off myoglobin. Myoglobin exists for the sole purpose to easily give oxygen away. So this is where that easy oxygen is going to come from. But this is going to go away in about 10 seconds. So now we're going to have to get more ATP. We don't have oxygen to make it. And so we're going to start taking phosphate groups from other molecules and putting them onto ATP. So remember, ATP is high energy. This is where our energy is going to come from. Our muscles need this in order to work. Once we use ATP, it's going to get turned into ADP. This is adenosine triphosphate. There are 3 phosphates on it. When we use one of the phosphates, it becomes adenosine diphosphate. There's only 2 of them. And so we need to convert this back to that. So normally we would do this through aerobic respiration or anaerobic fermentation. But this phosphagen system is going to provide us a kind of a middle ground. It's going to allow us to take phosphate groups off of completely different molecules and stick them onto ADP to get them back up to ATP. And so there's 2 enzymes that do this. There's an enzyme called myokinase. It's going to take 2 ADPs. So we're going to have an ADP and an ADP. If I take one phosphate off of this one and put it onto here, what does this one become? This is now ATP, and what is this? AMP, adenosine monophosphate. So I made an ATP. That's what I need. This is no less useful than that. Muscles can't use either of them. And so you could say, okay, instead of having 2 and 2, I'm going to have 3 and 1. Now I have one molecule that has 3, my muscle can contract. The other enzyme is creatine kinase. This is going to take a phosphate. This PI is a phosphate group. It's going to take a phosphate from creatine phosphate, which some people will abbreviate CP. And it's going to take it off that protein and put it onto ADP to make ATP. So essentially we're saying I have plenty of ADP. Where can I find a loose phosphate lying around that I can take it, stick it onto an ADP to make ATP? Myosin kinase is going to take one off of a second ADP. Creatine kinase is going to take one off of creatine phosphate. This here is the phosphagen system. So if we go back and look at this, so here we have, first 10 seconds is aerobic, then the phosphagen system is going to kick in, and it's going to get us for another 10 to 20 seconds. So we can go 20 to 30 seconds without having to get into the anaerobic fermentation. If you're sprinting, it's only going to last about 6 seconds. So you can imagine the faster you run, the faster you're going to use that up. So if you're running a marathon or something, you can make this last a whole lot longer than if you're sprinting. So this is just what we were talking about here. So myokinase is going to take one ADP, convert it into an AMP, take that phosphate that it stole from here, put it onto here. This is going to do essentially the same thing. It's going to take a phosphate off of creatine and put it onto ADP to make ATP. But obviously we need to be able to run for more than 20 to 30 seconds. So then we're going to have to switch over to anaerobic fermentation. There is glucose available in our muscles when we start. Most of the glycogen in our body is stored in our liver. But our muscles also have some glycogen stored in them. If we lose oxygen, we run out of oxygen in our muscles, our body can take that glucose and break it apart into 2 ATP and turn it into lactic acid. We're going to take 1 glucose to make 2 ATP and lactic acid. When we run out of glucose, we take the glycogen and break the glycogen up into glucose. Remember, glycogen was a big long string of glucose stuck together. And so we can take the glycogen, break it apart into glucose. You're in a process called glycolysis, like glucose, glyco, or sugar. Lysis is breaking. So breaking of sugar, glycolysis, and that's going to give us 2 ATP and lactic acid. So that's the glycogen lactic acid system. That's the entire process going from glycogen to glucose to lactic acid. We can only do this for about 30 to 40 seconds though, because we're going to run out of glycogen in our muscles. Our muscles are mostly myofibril. We don't have room to store a whole lot of glycogen. And so any additional is going to have to come from elsewhere in our body. So long-term ATP source would have to come from your liver. You have to get the glycogen from your liver. You'd have to start burning fats and things. That's why you can't run forever. You can't sprint forever. If you're going to run continuously, you have to run slow enough that you can breathe and replenish the oxygen as fast as you're running. And so if you're good in good shape, you're not going to require as much oxygen to run as someone like I would. And so you're able to breathe the same rate as me, but I'm burning more oxygen. And so I go into debt, where somebody else may be able to breathe as fast as they're burning. Question?
Why do they discourage like breathing really fast? You know, pass out.
I'm obviously not an athlete, and so I don't. I played golf in high school. that was just one half a second twitch. Does anybody? So you breathe very fast.
I think that just has to do with the amount of oxygen that's
Control it.
Probably not using the oxygen correctly that you inhale.
Maybe, yeah.